1 nanometer is 1,000,000 times smaller than a millimeter. This is the scale which Romanian scientists are working on to develop new mechanisms for breast cancer treatment. Using the pH gradient between healthy tissue and cancer tissue, nano-carriers will target 6 cells. The plan is to deliver specific breast cancer antibodies inside tumors. This approach can give hope even to metastatic breast cancer patients. Find out how this study is developing and when can we expect this cure to be released on the market.
What is the main objective of your project?
The project was a very generous one. We have tried to develop a new nanoparticle which has special characteristics, one of them being to target the tumoral tissue using a pH gradient, and the other one is to use different targeting molecules, in this case targeting specific proteins on the tumor side, in order to treat specifically the tumor cells and not to intoxicate the rest of the body.
How does the project plan to leverage the pH gradient between normal and cancer tissue to design and optimize nano-carriers for targeted therapy in breast cancer? And what specific characteristics will these carriers have to exploit this gradient effectively?
We had the benefit of using one of our very good chemical partners, it is the Institute of Petroponi in Iași, who developed a nanoparticle which is composed by different filaments of proteins which assemble and disassemble at certain pH levels. We have used a special characteristic of tumoral tissue which have a more acidic environment as compared with the rest of the body, and the design of the nanoparticle was as such as to produce an unravel of the molecules and release of the different substances in the tumoral tissue. So the configuration, the structural configuration of the nanoparticle changes as it passes from normal blood to tumoral tissue. And that is quite important. As an extra, we are using on the surface of the nanoparticles certain antibodies which do link to a specific protein which is overexpressed in certain breast cancer tissues.
Working with a diverse team and collaborating internationally with esteemed institutions like the Institute of Molecular Chemistry, Petroponi, and Oslo University Hospital, what advantages or benefits does this partnership bring to your team and your project?
It is very important to have partners who develop excellence in their level of research, because otherwise you cannot trust. It is very important because we all depend on the others in what they do. First of all, it was a privilege to work with Petroponi Chemical Institute because they are very much experienced in macromolecular structures and producing nanoparticles. They were able to produce the nanoparticles and all the structural modification that are asked by the project, and we have been very generous in asking lots of things from them, and they were able to develop nanoparticle and produce them for our experiments. So it was a great privilege, and it is not the first time we collaborate with them. It was also very nice to work with the Oslo Hospital research part, because they are highly experienced in assessing and evaluating the interrelation between nanoparticles and the cell membrane, which is very important at this stage, more so at this moment when we are trying to understand exactly what is the interrelation and how the nanoparticles are developing the results that we are seeing, which is an excellent result.
Could you share any preliminary findings from your research?
The project is now almost getting to close. So we have an extension of 4 months, but there's still a lot of work. We have results, some already published, and some results are still under the manuscript way. Soon we'll have submit one manuscript for consideration for publication. And we still have experiments involving transgenic mice for testing the efficiency of our product on animal model using human cell lines. And what we can say is that we start with several cell lines, both mice and mostly from human, test them in different way, different concentrations, and we found that one particular formulation of the nanostructure is more efficient in terms of release of what we want to test. So our results show that the molecule we are using, which one is binding to trastuzumab, increase the efficiency of trastuzumab therapeutic effect. So this it's very important for us to properly deliver a molecule we want for under pH gradient, and at the same time to have a better therapeutical effect.
Could you elaborate on the mechanism by which the nano-carriers aim to immobilize in tumoral tissue?
Nano-carrier is this nanostructure have outside linked trastuzumab, which is an antibody which specifically binds to a receptor from cell. But this receptor can be also in the normal cells. Sometimes it's overexpressed in tumor cells. So we target this overexpressed receptor and to deliver specifically charge as a correct place. So when the nanostructure will reach the tumor cells where the pH is different, we'll start to release what we aim to deliver.
How do you envisage the social impact of your project?
In many cases, it's happening that to appear resistance to treatment. So this project is aiming to restore the sensibility of trastuzumab in patients which already develop resistance. So we want an aim to allow the patient to continuously benefit from the therapeutic effect of trastuzumab in the case. Moreover, our results indicate in several cell lines that the therapeutic effect is enhanced. So we theoretically can try to use on mice for animal experiments, or maybe later in patients, to decrease the doses of chemicals used for treatment. In this case, also the overall toxicity will decrease and patients will have a better life status during this treatment.
What key takeaways would you like people to remember the most about your project?
It is a need for continuous dialogue between clinicians and researchers to try to solve the problems which appear during the clinical interventions. So maybe now it's not complete, maybe now we don't have enough for all the patients, all the cases, but there are still efforts to try to help the patients.
Thank you for watching Research in the Spotlight, a podcast for scientists and passionate science people made under the EEA Grants Research Programme by the Executive Agency for Higher Education, Research, Development and Innovation Funding as program operator. Until the next time, like and share this episode if you think science is our key to a better future.